All right, hello everybody. Welcome to micro seminar for the end of September. I don't know why Google Hangouts has zoomed in on my face so much, so I apologize. Normally I'm not so bug-eyed, but it's a pleasure to have Ben Roller with us today. He is a newly minted PhD from Tom Schmidt's lab at Michigan, which he went through a transition from Michigan State to Michigan during that time. Ben was in Daniel Bond's lab, of also previous micro seminar fame. Well, he's famous for a lot of reasons, but they have a great micro seminar that they presented a while ago at University of Minnesota. And Ben is also going to be moving on just in a couple of months to the ETH at Zurich, which is going to provide him with some exciting opportunities to study phenotypic variation within populations among single cells. So I'm going to turn it over to Ben. Just remember to tweet your questions to #USeminar or to the @microseminar handle, and we will get those questions into Ben when he's done. So go ahead, Ben, take it away.
All right. Sounds great, Cameron. Thanks so much for having me and inviting me here. It's an awesome honor to be able to give one of these seminars. So thrilled to be sharing my work with all of you guys today. Let's see if I can get my slides up and running. All right, are we seeing those come through?
That's all good.
All right. Well, thanks for attending my micro seminar today. I'm going to be telling you about my dissertation research in Tom Schmidt's lab that examines some links between bacterial growth physiology and genome evolution. So bacteria have a wide range of reproductive habits. For example, the maximum generation times recorded in the literature for a diverse collection of bacteria is massively variable. Here we're looking at a histogram of about 175 bacterial species, whose fastest doubling times can be as short as 10 minutes for Vibrio nitrogens, all the way on the left of this histogram, to more than 10,000 minutes for the fastest doubling time of the obligate intracellular pathogen Mycobacterium leprae. So while rapid reproduction is often thought of as a hallmark of bacterial life, the large variation we observe here indicates that factors other than rapid growth must influence bacterial fitness in the natural world. So I have used life history theory in my research to try to get a better understanding of the factors that influence reproductive variation in bacteria. Life history theory assumes that resources available to an organism are limited, and variation in reproductive habits are ultimately viewed as the outcome of natural selection acting to optimize reproductive success by investing these limited resources in different aspects of survival and reproduction. One fundamental idea from life history theory is that reproductive variation can be understood if one knows two pieces of information. The first is how traits which influence reproduction interact with each other, and the second is how these traits interact with the environment. I've attempted to examine both of these aspects of bacterial reproduction, and we'll tell you more about them today. So I will start by defining what a life history is for a bacterium. I'll then go on to describe how I have measured variation in life history among bacterial species. And then finally, I'll describe how a bacterium's genome content is related to its life history. So I guess this kind of brings the question fully back around. What precisely do I mean when I speak about applying life history theory to bacteria? An organism's life history is simply its pattern of survival and reproduction across the environments it inhabits. One example of differences in life history could be seen in mammals, which a lot of people know and have a kind of innate understanding of. Elephants have a life history pattern categorized by a long lifespan, slow development to sexual maturity, and low fecundity, or reproductive rate. So while these traits are fairly common for mammals relative to other animals, elephants kind of take these traits to an extreme. And there are other mammals who have other life history patterns, so showing that these life history patterns are clearly evolvable. Rabbits have a reproduction strategy that's much different from elephants, and these animals tend to have a short lifespan in which they reach sexual maturity early. They reproduce and make offspring at high rates. And while it's easy to observe these differences in reproductive habits for mammals in their native environments, it is much more difficult for us to do this for bacteria. So trying to just get an understanding of what bacteria are doing out in the real world causes a lot of problems. So what I'm going to do in place of showing directly what bacterial life history habits are is to show you a diagram of kind of conceptually what we think of as a bacterium's environment. Here I'm showing an organism's niche as a multi-dimensional description of the environmental conditions which allow it to persist. So here I've depicted three hypothetical bacterial species in terms of their pH, temperature, and resource requirements. The extent of environmental conditions where a species does not go extinct is shown by the white borders of this amorphous niche in this multi-dimensional space, which would in the real world be far more than just the 3 dimensions I'm showing here. The life history then for each of these hypothetical species is simply the pattern of fitness variation within the niche that the organism occupies. So we can look at the species on the lower left of this conceptual diagram, and we could describe its life history as being characterized by having a higher competitive ability at low resource concentrations and high pH, while the other 2 species are more competitive at lower pH and at higher resource availabilities. So now that we've got a little bit of a better understanding of just kind of conceptually what a life history is, I wanted to start to examine what factors tend to drive bacterial fitness patterns. So the rate and efficiency of bacterial growth have a large influence on a bacterium's competitive abilities. And the importance of these 2 features was demonstrated in a seminal paper in the early 2000s, which simulated heterotrophic bacterial growth in a range of environmental conditions. So these simulations involved 2 types of organisms: a slow and efficient organism that was innately expressing these phenotypes, and a fast and wasteful organism. And in the simulation, both resource availability and spatial structure were systematically varied, and the 2 types of organisms were competed across all of these different environments. Here I'm just showing the bacterium which grew slowly and efficiently, and it's in red, and its absolute fitness was positively correlated with both resource availability, regardless of the spatial structure of the environment. So that's depicted by an increase in the final population size as the resource influx increases there from kind of back left to front right. However, the relative fitness is really what's important when we're thinking about a competition between 2 organisms, and what I'm showing here is the outcome of competition in these environments. And this outcome of competition varied as a function of both resource influx, or the final resource concentration available, and the spatial structure of the environment. So here you can see that the outcome of the competition is ultimately depicted after one of these simulated species took over the simulated culture. And on the plot, you can see that the fast and wasteful organism always won at high resource concentrations and when the environment was homogeneous, or resources were distributed evenly across the entire environment. In contrast to that, the slow and efficient organism won the competition when resource concentrations were low, so all the way on the back left of the plot, and also at higher resource concentrations when the environment was patchy or heterogeneous. And really the take-home message from these studies was that when resources are privatized to a single species, whether it's by the privatization is by spatial structure or by low resource concentrations, these slow-growing and efficient organisms could win and avoid ultimately a tragedy of the commons, whereas the fast and wasteful organism would take over and outcompete the organism, but ultimately leave a lower final standing population size because of its inefficient growth. So ultimately, I view rapid and efficient growth as 2 different life history tactics. They integrate multiple underlying traits, which are shaped by natural selection to cope with these contrasting environmental pressures. In this case, the environmental pressures selecting for rapid or efficient growth is this resource availability gradient from scarce to abundant, along with the spatial variability of the environment. So there's a rich collection of evidence that I'm not going to show you here that goes beyond the simulation I showed, demonstrating that these features can and do select for rapid and efficient metabolism in designed laboratory experiments. However, there hasn't been a whole lot of evidence suggesting that these contrasting tactics are prevalent or important in natural populations or communities. And I've tried to use a comparative approach in my dissertation research to examine if organisms favored under these contrasting environmental conditions exhibit these rapid or efficient life history tactics. So the big question for me is, are these life history tactics observed in the real world? It's great if they're in simulations or in designed lab experiments, but I want to see, does this matter for microbes out in the natural world? And microbial ecologists have known for a long time that bacteria, which are prevalent in environments with low resource availability, have many different characteristics than organisms which are commonly found under high resource conditions. This plot is kind of a summary of what we know in microbial ecology about the spectrum of organisms that grow at low resource concentrations or high resource concentrations, and they're known as copiotrophs and oligotrophs. Copiotrophs have the ability to grow during resource abundance, while oligotrophs have the ability to grow during chronic resource scarcity. These are somewhat loaded terms in the literature, and it seems like they've been defined slightly different ways by many different camps in microbial ecology, but at their core, these categories merely describe where an organism has been observed growing, and they need not be mutually exclusive. It is frequently observed that many copiotrophic bacteria are capable of growing in low resource conditions, and in some ways vice versa. Some oligotrophic organisms are capable of growing when resources do become abundant. So one central goal of my work is to try to provide a useful way of moving beyond thinking of these categories as binary variables, and instead as existing along a spectrum. And ideally, I would like to do this by adding some evolutionary context to these descriptive ecological categories, and see if copiotrophy and oligotrophy explain this axis of life history variation that I've proposed. So what this boils down to is, rather than simply being a description of growth conditions, I put forward that copiotrophy and oligotrophy really describe the relative fitness of these organisms, and that copiotrophs have higher relative fitness during resource abundance, and oligotrophs tend to have higher relative fitness during resource scarcity. Furthermore, I suggest that copiotrophs achieve this higher relative fitness during resource abundance because of their rapid growth tactics, while oligotrophs tend to have a higher fitness during resource scarcity because of their inherent capacity for efficient growth. So to get at this question, the first thing we want to ask is, do copiotrophs employ rapid growth tactics? And there's a great deal of evidence that link copiotrophs to rapid growth, and I'll try to summarize some of that historical evidence quickly for you today. So a fundamental principle of growing bacterial populations was demonstrated by Jacques Monod in the early part of the 20th century, and he showed that growth rate depends on the concentration of limiting resource available to an organism growing in batch culture. So as the limiting resource becomes more abundant, the growth rate of a population tends to increase, and the number of doublings or divisions per hour then increases. This has been demonstrated for all types of bacteria and is one of the fundamental principles underlying chemostat cultivation, as well as a lot of other microbial physiology that some of us might take for granted today. The study of how bacteria increase their relative growth rate really arose from some of these early findings, and this work was done in the 1950s by the Copenhagen School of Microbiology with Ole Malot and some of the seminal papers coming from Motility Schachter and his group, along with Niels Kildegard. So the Copenhagen School of Microbiology really demonstrated that using experiments known as shift-up experiments. And what they demonstrated was that when you have an organism that's growing under unconstrained balanced growth conditions, so all aspects of its biomass are increasing at the same rate. Here I'm showing that as parallel lines that are increasing logarithmically with time. And what they showed was really that when you shift an organism from a relatively slower unconstrained balanced growth rate to a faster unconstrained balanced growth rate by shifting the medium, the first thing that happens during the shift up is that the transcription of the ribosomal RNA and an increased ribosome content is really the first response of these organisms to this increase in growth rate, or the ability to grow rapidly is really dependent on a higher ribosome content in bacterial cells. So rapidly growing cells are larger, and they tend to have more ribosomes than these slower growing cells prior to the shift up. After the shift up then occurs, overall measures of biomass tend to increase and become consistent with the rate of increase of ribosomal RNA. And then at a later time, the DNA content of all these cells starts to increase, and finally the cells get bigger. So over this time, cells are getting bigger and bigger, and finally they start dividing at a more constant rate. And we now reach unconstrained balanced growth again under a faster resource condition where all of these lines are again increasing in parallel, indicating that each component of biomass is increasing at a comparable rate. And I've used some of these fundamental principles of unconstrained balanced growth in some of the experiments I've done. So it's useful as a tool to demonstrate some of these physiological principles. So why do organisms need to increase their rRNA content when they start to grow faster? Well, one of the first things that was shown was that the protein synthesis rate per average ribosome in a faster growing cell only increases by a little less than a factor of 2. So the ribosomes themselves are already pretty close to maxing out their speed, and a cell, rather than trying to double its ribosome synthesis speed, instead just increases the content of RNA in a ribosomal RNA in a given cell, and in that way it increases the effective translation rate per cell. So that's ultimately the principle that underlies this increase in ability to grow quickly when nutrients become abundant. And this has been shown, you can show it numerically for E. coli. So if you have E. coli growing in unconstrained balanced growth at a doubling time of 0.6 per hour, it has about 10,000 ribosomes per micron cubed. But if you shift that E. coli cell up to double at a much faster rate, so growing at 2.5 per hour, you get a much higher ribosome content per volume as well as per cell. So you get some of these massive increases that you see in these much larger cells for E. coli are pretty commonly observed. But this rule also seems to be true for some oligotrophic organisms. So this work was done with Riccardo Cavicchioli's group on a model oligotroph, Sphingopixis alaskensis, which was isolated from most probable number cultivation, doing high dilutions over long periods of time to get these numerically dominant but slow-growing organisms into cultivation. And what they showed with this organism once they had it into pure culture was that when you shift it from essentially starvation, where it's not growing, up to its own relative maximum, which is much slower than E. coli, it also tends to increase the number of ribosomes in the cell. And if you look at it on a per biovolume basis, the numbers are very comparable to E. coli. So you get almost a tenfold increase, where E. coli you get a threefold increase in the number of ribosomes per biovolume. So this evidence almost suggests that RB2256, which is the Sphingopixis alaskensis strain we see here, might operate at a slower translation rate, or the ribosomes might actually be moving slower and synthesizing protein slower if it needs that big of an increase to increase its overall growth rate. And I'll come back to this a little bit later in the talk. So here we just see the overall pattern that rRNA content enables rapid growth. And next I'll show you a little bit about how the ribosomal RNA genes are organized in a bacterial genome. So in nearly all bacteria, the rRNA genes are co-transcribed together in an operon, which is named the rRN operon. So simply leaving off the A, we get rRN. And this typically includes genes for the 3 ribosomal RNA subunits of the ribosome, as well as a variable number of tRNA genes depending on the species or strain you're looking at. But this canonical structure is pretty unvariable across species. There's some variation in it. Some organisms don't have this 5S gene at the end of the operon. Some organisms might have an insertion somewhere along this place. But typically you see this general overall conserved structure. So some of the original work looking at ribosomal RNA operon content in bacterial genomes was done by Tom Schmidt's lab, where I did my PhD in the late 90s and early 2000s. And they demonstrated that the ribosomal RNA gene content of bacterium's genome is predictive of how rapidly those organisms are going to respond when resources become abundant. So what they showed was that with a simple soil plating experiment was to take a soil bacterial community, dilute it, and plate it on nutrients, so you're going from a relatively nutrient-poor condition to a relatively nutrient-rich condition, and just observe the appearance of colonies over time, and then measure how many ribosomal RNA genes are present in those organisms' genomes. For colonies that appear quickly, so in less than 48 hours is the cutoff they used, or colonies that appear much later on, sometimes for this category much later than 150 hours after the initial incubation was started. So what they showed was that these fast-appearing colonies tended to have many more copies of the ribosomal RNA operons in the genomes, and colonies that grew slowly or appeared late tended to have many fewer ribosomal RNA operon copies in their genome. And this really provided some of the first evidence that you can use a genome feature to make predictions about an organism's ability to respond when a nutrient changes. So what I've done is try to take this a step further and use ribosomal RNA operon content to look at the relationship between maximum growth rate and RN copy number across a wide range of bacterial genomes. So I've done this for 8 bacterial strains that I've cultivated in the laboratory and span the spectrum from 1 to 13 RN copies. And this covers most of the range known for bacterial species. There are a few that have 15 rRNA genes in their genome, but I wasn't able to get those to grow under the same conditions as we had here in lab. But if we look at the maximum growth rate for these 8 strains across any environment that these organisms are able to grow in, we see this overall general positive relationship where there's an approximate doubling of the maximum growth rate with every doubling of ribosomal RNA operon copies these organisms have in this genome. So we're seeing this strong overall trend for the organisms that I was able to grow in lab, but what about kind of the rest of the bacterial world? What does that look like? So here I'll reuse some of the data from the histogram I showed previously, and we can look at the number of ribosomal RNA genes and the maximum recorded growth rate under any condition for all of these about 180 now bacterial species. So here each dot again represents a different bacterial species that we know its copy number, and there's some evidence for its maximum growth rate in any environment in the literature. And we see the same trend that we saw with my data indicated in red, that on average, you get an approximate doubling of maximum specific growth rate in any environment with the doubling of RN operons in the bacterium's genome. So while doing a simple regression, like I have in this analysis, is a reasonable way to approach the data on first pass, we also know that not all of these species shown here are independent of one another. And in fact, some of the organisms are more closely related to each other than others. And we need to correct for this non-independence when we do statistical analyses. So I'm doing these what's called phylogenetic regression approaches throughout this talk, and I'll introduce the concept here on this slide. But really what we're talking about is the organisms on a phylogenetic tree that are more closely related to each other are more likely to have similar ribosomal RNA operon copy numbers. So in this mock example here with 5 species from the data, we see that the most closely related organisms share the same ribosomal RNA operon copy number. But as we go to more and more distantly related organisms or pairs of organisms, there's more variation in how close the pairs of organisms match their RN operon copies. So rather than just look at a few of these data points, here I'm just showing the species names, and you can see that within, in general, a genus, we get very similar groups, but as you start going beyond the genus level differences, we get more variation. But if we look now at what we know for all bacteria, so here we're going to look at thousands of bacterial genomes and where we know the ribosomal RNA operon copy number, we start to see some really broad and big patterns. So what I'm showing here on this tree on the right is a heat map. So essentially the number of ribosomal RNA operon copies is indicated by the darkness of the color. So low RN operon copy number is indicated by the darkest red, whereas the highest RN copy number is indicated by the brightest yellow. And what we see is that there are some distinct bands where clades of organisms all tend to be high copy or low copy, especially at the bottom of the tree where there are some gamma proteobacteria, and in the middle of the tree in the firmicutes where we get some really high RN copy number. But I think one interesting thing across this whole broad tree is that there are a lot of swapping between high and low copy at a very high resolution within the tree. So at the tips of the tree, we get a lot of switching from red to yellow to red to yellow as we look at the tree. So overall, there's a lot of kind of evolutionary plasticity in this trait of ribosomal RNA operon copy number, and we ultimately need to correct for that a little bit of shared evolutionary history to make sure it's not influencing our regression. So on the plot that I'm showing here, the dashed line is the phylogenetic regression line, which takes into account this non-independence between data points, and we see that there is this doubling of growth rate with doubling a copy number that still exists, and overall our conclusions aren't changed in this case. So ultimately, with this data, I've showed that Copiotrophs employ rapid growth tactics, and now I wanted to move on to ask, do oligotrophs tend to employ efficient growth tactics? So here, I'm defining growth efficiency for heterotrophic bacteria as their carbon use efficiency, or the number of offspring that they can produce per unit of resource consumed, which would be the growth efficiency measure, is really equivalent to the amount of biomass that ends up in, or the amount of carbon that ends up in biomass that they're growing on, relative to the total amount of carbon that these organisms are utilizing. So under unconstrained balanced growth conditions in the laboratory, we can show that this overall relationship is true, that growth efficiency is equivalent to carbon use efficiency for aerobic heterotrophs. And in order to really establish that this is the case, we needed to show that the environmental drivers of growth efficiency variation can be controlled in laboratory settings. So here we know that an organism's carbon use efficiency is highly dependent on environmental parameters. So if we're going to try to compare across organisms, we need to control for these environmental factors. So we need to measure all organisms under the same resource availability and under the same resource that has the same amount of energy available to it in terms of its oxidation state. So ultimately, there's kind of a conceptual model of the experiments we want to run. So in order to control for the environmental drivers of growth efficiency variation, we need to compare our copiotroph and oligotroph organisms and everything in between under the same limiting resource concentrations and under the same carbon resources that are available to it. So ultimately what we would be doing in this case with a model copiotroph and a model oligotroph would be to measure these organisms in the red circle where they're exhibiting the same environmental conditions and really ask, is there a difference in the population's mean growth efficiency between or across all of these different populations? So what I did was grow this series of 8 organisms that I highlighted previously under unconstrained balanced growth conditions with succinate as a sole carbon and energy source, and the rest of the medium was just mineral salts. So organisms need to make all of their biomass out of these succinate molecules, and ultimately we measured how much of their biomass ended up or how much of the carbon in succinate that they used ended up in biomass, and how much total was used with a few different methods. We used radio labeling with tritiated leucine and oxygen sumption and converted those to carbon units. And then we also directly measured carbon use efficiency with 14C radio labeled substrates as well. And again, the growth efficiency is equivalent to carbon use efficiency once you get to these unconstrained balanced growth conditions that I'm showing. So our results, when we look at the 8 organisms I showed previously, with both of the different methods we've utilized, show that on average, for every doubling of ribosomal RNA operon copy number in a bacterium's genome, we get about a 3% decrease in carbon use efficiency for each of these doublings. So just like growth rate was logarithmically related to RN copy number, we seem to see that for every doubling of copy numbers, we get the same unit decrease in efficiency. So this log2 relationship between RN copy number and rate and efficiency jumped out to us as interesting, and that we had some statistical support through AIC analyses showing that that is the best fit to the data. And we ultimately can show that these relationships are inverted to one another and suggest that maybe this ribosomal RNA copy number gradient is a good proxy for where an organism lies on a trade-off between rapid and efficient growth. So low ribosomal RNA copy number bacteria tend to grow more slowly, but they also tend to use carbon more efficiently. So this suggests that there may be some evolutionary trade-offs going on between an organism's growth rate and growth efficiency. So here I can show that data directly. So looking at the maximum growth rate against the carbon use efficiency for the 8 strains we've measured here, using a few different phylogenetic regression techniques. One assumes no error in both in the x variable, but we know that's not true. So we've done the ranged major axis regression as the dotted line, which is the best fit to our data, where both variables are experimentally measured and potentially have error in them. And what we can show is that there is this overall negative relationship among all the species in our data, and it can't be explained by shared evolutionary history. That there's this negative relationship between an organism's maximum growth rate and its carbon use efficiency. And that the organism's place along the spectrum can really be predicted by the rRNA gene content within its genome. So one question we had was, how might these oligotrophic organisms grow more efficiently? What are the physiological mechanisms that allow a copiotroph to grow more efficiently than an oligotroph? And we used some of the radiolabeling data that we had, and also used some previous data that's been collected in the lab and published by a graduate student that preceded me in the lab, Les Dethlefsen. And what Les showed was that when you look at bacteria that range across this ribosomal RNA copy number spectrum, they tend to have a higher translational power, and the ribosomes themselves actually are synthesizing protein at a faster rate in high copy number bacteria when you measure them under the same environmental conditions as low copy number bacteria. So this, to us, suggested that maybe this trade-off between rate and efficiency, if it exists, could be due to differences in ribosome phenotypes, and maybe it's the rate and efficiency of protein synthesis that are really driving the overall rate and efficiency of growth. So with Les's work, we've got one half of the data showing that with the phylogenetic regression, there's a positive, significant relationship between the ribosome synthesis rate and RNA copy number. And my data with tritiated leucine and oxygen consumption, shown here as the protein yield, or the amount of protein an organism can make per unit of energy consumed, is again showing a negative relationship. And this indicates to us again that this hypothesis that the ribosomes might be driving a potential rate efficiency trade-off across bacteria, our data is at least consistent with that idea. But again, we can't prove that this trade-off exists with comparative data alone, and we'd have to do some sort of experimental evolution experiment to really get at if RNA content really is predictive of a trade-off, and if that trade-off is being driven by ribosome phenotypes. So with that data, I think we've got some decent evidence to suggest that oligotrophs do tend to employ efficient growth tactics and grow more efficiently than copiotrophic organisms. And next, I wanted to ask the question, does ribosomal RNA operon copy number really a proxy for an organism's place on the spectrum from copiotrophy to oligotrophy? So the approach I used to get at this question of is copy number a proxy of the life history spectrum is to infer life history traits from genomes. So we can, we've got about 1,000 genomes where we know ribosomal RNA operon copy number, and we can take advantage of all this data by inferring some of their physiology from their genome and trying to ask questions at this broader level with much more data than we're able to in the lab. So again, inferring life history traits and testing if these postulated life history traits have the expected relationship with log2 ribosomal RNA operon copy number as we would expect a priori. So the data set, again, I'm using is over 1,000 bacterial species where we have complete genomes with known ribosomal RNA operon copy number, and we've got Kegg's annotation of the orthologous genes in the genome to make predictions and inferences about the physiology of these organisms. The first place we wanted to ask a question was within chemotactic motility. We know that chemotactic motility is an expensive way to find resources in the environment, and bacteria tend to use this to sense and move toward high resource concentrations. So in the canonical example of chemotaxis for E. coli, it's kind of a random biased walk is how it's described. So bacteria like E. coli will operate their flagella and have the ball working in concert to move in a straight direction, But this straight direction swimming only happens for a short amount of time, and the flagella then stop all beating together, and the organism tumbles and reorients itself within the environment. And chemotaxis really allows these organisms to direct their swimming up a resource gradient, which is examined here, or shown here in this diagram, by leading to more biased and increased straight swim distances when the organism senses an increase in the resource concentration from the previous position to the final position. And over time, this bias of increased straight swimming towards increased gradients leads to an overall movement towards a higher concentration of resources. And what we expected was that this might be advantageous for copiotrophic bacteria, but it could be extremely expensive to run and may be disadvantageous for oligotrophic bacteria. And this is because there's a high energetic cost of chemotaxis, which depends on many variables, including the nutrient concentration in the medium itself, as well as the relative gradient, the viscosity of the environment, the cell size of these organisms, and the swimming speed of the organisms. So for example, if you look at E. coli in rich medium growing in the laboratory and there's a gradient within that rich medium, E. coli doesn't spend a whole lot of its energy budget on chemotaxis, and maybe 2% of all ATP end up going towards fueling chemotactic motility. But if you look at, say, a Vibrio species in the ocean, and you can make some assumptions of what its environmental parameters are, the Vibrio species might be using as much as 50% of its energy budget to reach these higher nutrient concentrations. So clearly there are some costs towards chemotactic motility in natural environments. So again, we're really asking the question, are oligotrophs more likely to be immotile or not to use chemotactic motility? And this is also due to the fact that oligotrophs tend to have very small cell sizes, which make motility even more costly than it would be for a larger Vibrio cell. So our postulate, again, is that Copiotrophs chemotax to exploit rare resource patches, while oligotrophs would wait for diffusion to bring resources to them. And we inferred chemotactic motility now for all 1,000 genomes in our database and compared that to ribosomal RNA operon copy number. And what I'm showing here is a plot of the logistic regression we can do both taking into account phylogeny, which is the dashed line, as well as ignoring phylogeny, which is the solid line. And what we see here is I'm plotting each bubble represents all of the organisms for that given ribosomal RNA operon category, and the size of the bubble is the number of genomes in that RN copy number category. So any given genome would be a 1 or a 0, indicating the presence or absence of chemotactic motility genes, and the ultimate ratio of organisms, or the probability of organisms in that category, is plotted on this graph for ease. So we fit the logistic regression to the underlying binary variables of yes or no for chemotactic motility, and what we see is this positive relationship. So high copy number organisms are more likely to encode chemotactic motility, and this can't be explained by the shared ancestry of high copy number organisms. So indicating that there is some adaptive advantage, it seems like, for chemotactic motility in copiotrophic organisms. Here I can show you kind of an overall table of all of the different hypotheses that we're going to test. And again, we expected chemotactic motility to have a positive correlation with copy number. We saw that, and it couldn't be explained by phylogeny. Next, we wanted to ask about genome streamlining. So this is a topic I know near and dear to some other of the micro-seminar participants' hearts. And genome streamlining, the overall idea is that oligotrophic organisms are observed to tend to have smaller genomes than other non-oligotrophic organisms. And it's thought that this might be due to the selection acting to minimize reproduction costs. This is a separate hypothesis than what's known for obligate intracellular bacteria, which also tend to have small, reduced-size genomes, because genome streamlining really implies selection for minimizing costs, whereas for obligate intracellular bacteria, it's often thought that drift is the cause of the loss of a lot of the genes leading to these small genome sizes in those organisms. So here we're asking if there's a selective benefit for having small genomes in low RN copy number bacteria, and we predict, again, a positive relationship between genome size and RN operon copy number. And we do tend to see this overall effect, but it is a little bit smaller than we thought it might be. So when we account for phylogeny in the regression, we tend to see, and I notice there's a slight error on the slide, but about 0.66 megabase increase in genome size from an average 1 copy number organism to an average 15 copy number organism. So there is this positive relationship, but it's not as extreme magnitude as we thought it might be. And again, we know that rRNA genes do take up more of the genome, so we tried to do some calculations to see, can this whole pattern be explained by just the increased rRNA gene size? And no, it cannot. So this relationship does seem to be true, but again, kind of weak and not particularly strong across all 1,000 bacterial genomes we're looking at here. To look more directly at if genome streamlining is playing a role in the evolution of oligotrophic genomes overall, we wanted to ask a specific question inspired by Paul Carini's research in Steve Giovannoni's group about genome streamlining in the thymine biosynthesis pathway. So thymine is a necessary cofactor for several core TCA cycle reactions, as well as the Kelvin cycle and pentose phosphate pathway. So it's a cofactor that seems to be essential for a lot of kind of basal metabolism and microbial life. And we wanted to ask and follow up on Paul's insight that oligotrophic organisms often tend to lose genes in this pathway and rely on environmentally available thiamine or its precursors and just recycle that rather than synthesize it all themselves. So that might allow these organisms to lose genes in the biosynthesis part of this operon and or the biosynthesis part of this pathway while maintaining the recycling portion of the pathway. So the question here is really, do low rRN genomes possess fewer thymine biosynthesis steps? And what we see when we look at the full thymine biosynthesis pathway with a Poisson regression here indicating the number of encoded steps on the y-axis, and the darkness of each color represents how many genomes tend to have that many steps at that copy number category. And what we see is this overall positive relationship where low copy number genomes tend to encode fewer thymine biosynthesis steps, and high copy number organisms tend to encode many thymine biosynthesis steps. And on average, if you look at the relationship with phylogenetic regression, we tend to see this increase of about 3 steps from an average 1 copy number organism to an average 15 copy number organism, indicating that, yeah, there's significant gene loss in this pathway along this copiotroph oligotroph spectrum for all bacteria that we know of. And we can look more specifically at what genes are lost in the pathway, making predictions about that the genes that are essential to de novo synthesis might be lost, whereas the recycling genes might be retained in these oligotrophic genomes. And that's really saying, are the absent genes randomly distributed in the pathway in low copy number genomes, or are they more conserved under the de novo genes only? So we can split up this thymine biosynthesis pathway into steps that are essential only for de novo synthesis. There's also one gene that's poorly annotated and is only present in about 40 of all 1,167 genomes we have. So we've ignored it for this amount of time and asked, is the gene loss happening in these 8 de novo synthesis genes, or is it happening in the 3 reactions that are essential for recycling exogenous thymine or its precursors? And what we tend to find is that we can account for all of the gene loss that we found when we look at the whole pathway within the 8 reactions that are exclusive to thymine biosynthesis. So this implies that, again, oligotrophs aren't just losing a bunch of genes at random through drift, but instead are there's selection acting to minimize reproduction costs when they can utilize exogenous thymine from the environment. So here, again, just showing it along this table, we tend to see the expected correlations between genome size and thymine biosynthesis genes with ribosomal RNA operon copy number, and those are significant when including phylogeny in these regression models. We also asked a few questions about PTS transporter richness. So the PTS transporter system is a specialized thing for sugars and tends to operate best when there's a high extracellular concentration of these sugars. So we asked and hypothesized that these transporters, which are specialized and only used under high resource concentrations, might be more common in high copy number organisms. And we do see the relationship between PTS transporter richness and rRNA operon copy number, but only when we exclude phylogeny from the model. And it seems to be that PTS transporter richness is only very high in a few clades of organisms. There's a few clades of gamma proteobacteria and a few clades of firmicutes that have high copy number and high PTS transporters, and that drives most of the signal in our data. So when we include phylogeny in the model, we only see the signal kind of being wiped away, and there's no effective difference, even though the p-value is significant, between genomes. So indicating that there's some evidence for the hypothesis, but it could be explained by phylogeny alone or shared ancestry of this PTS transporter phenotype. Finally, we asked if autotrophy was related to ribosomal RNA operon copy number in genomes, and we hypothesized this because we thought it might alleviate organic carbon competition, and we expected a negative correlation between autotrophy and copy number. And we do observe the correlation again, but it's mainly being driven by the cyanobacteria, which make up a large fraction of the number of autotrophs in our data set. So ultimately, when we include phylogeny in the model, we no longer see this relationship, indicating that the pattern could be just phylogeny alone and not due to a significant adaptive advantage for these oligotrophic low copy number organisms to be autotrophs. So overall, with all the genome data that I showed you so far, I think we have some good evidence that copy number is a proxy for this copiotroph-oligotroph spectrum. Between the physiological and genomic data, we're seeing this relationship hold up. And ultimately, we're seeing that this prediction that we have for where an organism might lie within its niche and could be predictable by the ribosomal RNA operon copy number in its genome. And specifically, we're saying that the RN copy number of an organism might lie parallel to its place on the resource availability spectrum. So oligotrophic organisms are more fit and have low copy number, and they're more fit under low resource concentrations, where the copiotrophic organisms that are more fit under high resource conditions tend to have more copies of these ribosomal RNA genes. So finally, I wanted to ask, kind of overall, does an organism's genome content covary with its ribosomal RNA operon copy number? So now in this case, we're going to be looking at some ordination plots where we compress an organism's overall genome content, so all the orthologs or all the pathways in its genome, into one data point, and we ask, is that organism's combined set of pathways more similar or less similar to every other genome in the data set? Using ordination techniques. So here we're just using non-metric multidimensional scaling to do this ordination, and what we see is a clustering of genomes by their phylum level membership. So genomes within the proteobacteria have points that are more close in NMDS space than they do to actinobacteria or firmicutes, indicating that there's a lot of phylogenetic inertia to an organism's genome content. So proteobacteria genomes look more like proteobacteria genomes than any other phyla, and firmicutes as well. But the interesting thing to me when we look at this plot, and rather than color by phylum membership, is to color by the organism or genome's rRNA operon copy number, is that we see very similar and kind of parallel relationships within each of the three major phyla in our data set. So here we're seeing an increase in copy number indicated by an increase from dark to light coloring for a genome, going up NMDS axis 3 for all three of the phyla in the analysis. So to me, this indicated that maybe the genome content of copiotrophic bacteria is more like the genome content of other copiotrophs than it is of oligotrophs. And to really test this hypothesis, we needed to control for phylogeny when we do these multi-dimensional ordination style analyses. So we used phylogenetic PCA to collapse the phylum, or to collapse the genome content for all of these 1,000 bacterial species. And again, we're combining all the different variables, so all the different pathways or orthologs within a genome into one data point and looking at the comparison across species. And what we see when we do that and explicitly control for phylogeny is still this overall pattern of genome content similarity being more similar for low copy number organisms and high copy number organisms, and this kind of overall gradient of genome content similarity across the major PPCA axes in our data set. So this is a little bit hand-wavy just looking at the data, but what we can do is take a PPCA axis and ask, does that axis score for each of the genomes correlate with RN copy number in a regression-style analysis, so axis by axis we can go through it. And what we see when we do that is that many of the top axes in these ordinations are all correlated with ribosomal RNA operon copy number, indicating that genome content, whether you think of it in terms of modules or pathways on the left, or in terms of individual orthologs on the right, are all highly correlated with ribosomal RNA operon copy number. And we've also done this in a multivariate way, so rather than looking at each axis independently, we can look at all 50 of the top axes in a PPCA plot. And we also show that, again, there's this significant relationship with copy number. And we've done a little bit of follow-up work with this, looking at other axes of a microbe's niche, and we don't seem to see the same relationship of an organism's temperature niche or oxygen preference niche dictating genome content as much as ribosomal RNA operon copy number does. So that was kind of the last data slide I have, and it really shows that rapid and efficient growth tactics as measured or approximated by RN copy number does explain some similarity in genome content across all bacterial species. So in summary, I've shown that oligotrophic bacteria tend to have few RN copies and grow more efficiently than copiotrophic bacteria. Also, that RN operon copy number systematically varies with rapid and efficient growth tactics. And finally, I've shown that life history evolution has led to genome content similarity among distantly related species. So in the future, we hope to use this to discover some new life history adaptations in bacterial genomes. So we came at this question with some a priori hypotheses of what we thought might be adaptive for an oligotroph or a copiotroph, but we can also use these ordinations to ask what genes are truly driving these differences between low copy number and high copy number genomes. So here we'd be looking at a loading on a PPCA axis, and some of the highlights from what we've kind of found and some of the new stuff we're doing shows that osmoprotectants such as glycine betaine are more likely to be synthesized and transported by high copy number bacteria, and low copy number bacteria just don't seem to have the mechanisms to cope with osmotic stress that high copy number bacteria do. We've also shown and got some hints that are pretty exciting that the type 3 secretion system, which is often used in interactions with eukaryotic cells, and so between bacteria and eukaryotes, and that these interactions may be mediated by an organism's life history, and that high copy number copiotrophic bacteria might be more likely to interact with eukaryotic cells using this type 3 secretion system. Finally, I think our work really can shed a light onto some of the work done and shown by Kristen D'Angelis in her seminar, that we can test ecological predictions for bacterial community composition using ribosomal RNA copy number. So those methods briefly just use a known bacterial tree to predict the copy number of unknown OTUs in a bacterial sample, and you can really look at a community copy number distribution and ask, does an experimental treatment shift the copy number of the average organism in the community, or the distribution of copy numbers in the community, from one treatment to the next? And that could potentially help shed light on how communities of organisms really are assembling. So with that, I'd like to thank a huge number of people from the Schmidt Lab for their ideas and kind of developing this project over many, many years in the lab, especially Tom, Zaraz Lee, Stephanie Eichhorst, Les Dethlefsen, Brad Stevenson, and Joel Klappenbach have all really built the base of this project. And I'd also like to thank the funding sources. And with that, I'll take any questions.
All right. Awesome. That was a really cool talk, Ben. Do we have any questions from within the seminar right off the bat? We have a couple on Twitter from John Badalamente from a long time ago, from even before the talk started. John asks, if I see tandem rRN duplications, triplications in iron reducers, is this real or lab selection?
Yeah, that's a really good question, John. Thanks for asking. So the evolution of ribosomal RNA operon copy number does seem to happen through duplications, so it certainly could be a possibility, but I think what you would really need to do to ask if there is selection that has led to your duplication or not is to get some genomes that are very close neighbors to your organism and ask, is there an ancestral architecture of these RN copies near each other or not, and kind of do a phylogenetic comparative approach in that sense and ask, is this duplication event seen in other organisms that haven't been selected for or not? and is it just a product of this organism's evolutionary history?
Okay, he has a built-in follow-up as well, which is, are bugs striving for faster growth rate, better adaptability, or both?
So that's, I guess what I would think of what he means as adaptability is just an ability to respond when environments change. And I think those two things go hand in hand from some of the data we've shown, that an organism with a more rapid growth rate is more likely to respond and adapt to environmental change, whereas oligotrophic organisms that are great at using small amounts of resource might just specialize in that condition and not really care what happens to the environment around them and eke out this living kind of grinding it out day to day to day and not care or adapt or change when the environment changes.
Yeah. Okay, we've got a question from Dave Baltris. So what's up with the tips of the trees where rRNA copy number jumps around a bit? Is this indicative of ecological differences potentially?
Yeah, I think that really is some of the strongest evidence we have. The tree I showed you doesn't have any obligate intracellular bacteria, but we also see evidence for that with these obligate intracellulars, where we have a clade of gamma proteobacteria like E. coli that all have 7 copies. And some obligate intracellulars have evolved within that clade, and they've all lost kind of this ancestral high copy number and are now 1 or 2 copy number organisms. I would argue, because we see it a lot across the tree, so it doesn't just happen once where there's gene loss, but kind of repeatedly we see these losses where there's some differences that I would guess it is a signal of ecological specialization and not just a product of drift or some random event of loss.
That's neat. All right, Kristen DeAngelis. First of all, she says she was really enjoying your seminar, and secondly says, does lack of osmoprotection contradict with equilibrium growth strategy characteristic of oligotrophs? I think I know where she's going with this. If you're trying to stay, if you're trying to hang out in a steady state and grow kind of slowly, if you have a lack of osmoprotectants, doesn't that make that a more difficult thing to do? Kristen, you can correct me if I misinterpreted that, of course.
Yeah, no, I think that question is a bit of a surprising one, and we're still trying to wrap our heads around it too. So yeah, my initial thought wouldn't have ever been to look at that hypothesis, but it's just kind of what we see in the data. And I think there are 2 potential explanations. One could be that oligotrophs just don't use glycine betaine but use other osmoprotectants or have other strategies besides that to deal with osmotic stress. So maybe the one kind of well-known pathway we know is biased towards copiotrophic organisms. There are other osmoprotection strategies out there and we just don't know them, so we can't ask questions about them. But another potential thing might be that oligotrophic organisms just might not be adapted to deal with it, and they've lived in the bulk so long that they just hope that they have a high enough population density when that osmotic stress happens that enough of them survive to live on. And I guess I would be less likely to guess that hypothesis is true, and it's more likely that there's just some unknown mechanisms. But this glycine betaine thing is a very interesting thing we're interested in following up on.
Yeah, one thing I wanted to add to that, Steve Giovannoni's lab showed a couple years ago that SAR11 actually oxidizes glycine betaine as an energy source rather than produces it. So, one of the things that this is making me think about is there any way that the directionality of these pathways might be playing into their presence-absence and the oligotrophy-copiotrophy argument? Because, you can look at presence-absence, but, in SAR11 it looks like the pathway is going the other direction.
Yeah, absolutely, and that's a whole other, I think, difficulty when you're trying to infer physiology from genomes, is to know what direction is a pathway moving and when is it operating, and is the presence of a gene a great indicator of this physiology. So I think, yeah, it's always something you have to keep in mind and worry about as we're thinking about these questions. But I think what we're really doing with some of these methods is to generate hypotheses and not really test them. And I hope some of this work can inspire other people to go in and start knocking genes out, manipulating organisms, and seeing, do these organisms really behave this way, or is it something that comes out of just looking at it from a 10,000-foot view?
Yeah, yeah. We've got a question from Stuart Jones. Does mineral nutrient economy, for example, phosphate quota or phosphorus quota, correlate with carbon use efficiency or rRNA copy number? And if so, in which direction?
Oh, that's a great question. So he's asking if, like, phosphorus use efficiency is correlated with carbon use efficiency.
Yes, or rRNA copy number.
Yeah, so I think, kind of from my point of view, I think they should be correlated with each other, and I think the environment really does drive a lot of these patterns. So if you look at a single organism, it doesn't have one carbon use efficiency, but that CUE is highly variable depending on the environmental parameters. And I think when an organism tends to have a high CUE, it also tends to have a high phosphorus use efficiency as well because the environment is rich and allows it to grow a little more rapidly. So I think if you're seeing these things in a natural system, and you see a high carbon use efficiency, the first thing I would think of is that the environment is allowing all these organisms to reach a more unconstrained or balanced growth condition, and the organisms are more happy. But if you're seeing these differences between organisms, yeah, that's where it starts to get really interesting. Is PUE still high but relatively lower in Copiotrophs? Is still a really difficult question to ask.
I have a follow-up to some of these new directions you're going in. Have you looked at regulatory genes by any chance and checked out any trends in this sort of relationship with regards to that? I know that there's some preliminary evidence that there's much lower sigma factor content, for example, in streamlined genomes. But what about other types of regulatory components?
Yeah, that is a question we haven't yet gotten to, and I think it's a beautiful question, and I would agree with you that I would think the genetic repertoire of ability to respond to an environment, if this hypothesis is true, that oligotrophs just don't have the responsive capacity that copiotrophs do, we would expect that these gene regulatory switches would be less abundant in their genomes as well. So yeah, absolutely I agree, and I think it would be a great direction to look at as a function of copy number. So I think you published a little bit on that. I know I'll have to dig into your papers a bit and see what you guys use for guidelines for sigma factors and other regulatory elements.
You need to talk to Ben Temperton. He did some really cool analyses on that. Okay. Are there any other questions for Ben? This has been a really nice question session, actually. Anything else that's coming up in anyone's minds? Well, it's food for thought for sure. This is really stimulating seminar. I know a lot of people will probably be using this in their classrooms. I know I probably will next semester. Ben, how can people get a hold of you if they have follow-up questions?
Yeah, so please get in touch via Twitter or email. My email is my last name, Roller, be@msu.edu, and yeah, I'm happy to talk offline or keep the conversation going, so please send them my way.
Okay, wonderful. Well, thanks again, Ben.
Yeah, absolutely. Thanks for having me.